morning/afternoon, everyone. My name is Verena. I'm from the Department of Biodiversity and Conservation Biology at the University of the Western Cape in South Africa. Just before I kick off my talk, I just wanted to say that it has been a really fantastic experience to see all of the wonderful work being done on jellyfish throughout the last couple of days. It's great to see more and more regions represented, and also to see more and more taxa being studied. But in as much as we are seeing an uptick and an increase in jellyfish research across the world, unfortunately, there are still many regions that are sorely underrepresented. Africa is one of those regions, notorious for being underrepresented in several domains, with global studies often only having one or few samples from Africa. For example, the study by Aboud, which was a truly global genetic study, but only encompassed one sample locality from Africa. The entire African continent was represented by only one sample. This is problematic. But thanks to the advocacy that Professor Mark Gibbons has been doing over the last decade, probably much longer than that, jellyfish has been added to the sailing orders of a couple of research efforts, particularly the Dr. Fridtjof Nansen, which embarked on widespread collections across the entire African coast, collecting not just jellyfish, but lots of other taxa along the way. This expedition spanned the west coast of Africa as well as the east coast of Africa, and probably encompassed the most widespread collection that we've seen in recent years. And so you can imagine how excited we were to get our hands on a lot of this data and a lot of these specimens. So I was lucky enough to be able to work with a lot of the West African samples. And of course, I couldn't look at everything. And so what I did was I randomly selected samples from each leg of the cruise and just looked at what I was seeing, essentially looking at patterns of diversity, uncovering some genetic diversity or taxonomic diversity. I looked at a lot of taxonomy, but I focused a lot more on the genetics. So I did a lot of barcoding, not just CO1, but I looked at a range of different gene regions. And I want you to brace yourselves for what comes next. I found some really, really cool things already, although I didn't have such large sample sizes. We saw some really interesting things. Lots of atypical patterns emerged in Africa, which I'm going to cherry pick a few of them. I'm not going to present all of them, but I'll present some of the things that I found really, really cool. So I'm going to start with the Otolla. So we know that the going theory is that with increasing depth, we typically have increasing gene flow. While quite interestingly, the Otolla species that we recovered, or taxa that we recovered, showed instances of sympatry actually with Otolla van Hoefene and Y Valley present on the West African coast and occurring in sympatry in a couple of the samples that we analyzed. And although this is not the best tree, this is a consensus tree. I just wanted to show you some of the diversity a little bit more visually. And you can see that from this tree alone, there is a little bit more structuring even that might exist, which we can explore further in the future. So this was really cool for me to see already that a deep ocean species is showing so much variability. Fast forward to the Pelagia. We also recovered quite a number of Pelagia samples from the West African coast. And we also saw some very interesting patterns. These are not just West African samples. These encompass samples from Ale, Stopa, and Miller as well. But I'm going to point out a couple of interesting things. The cluster you see over there is mostly Southern African samples, so samples collected from South Africa. And interestingly, although it's not maybe presented so well here, and maybe you can't see it so well, they actually formed a cluster with a number of haplotypes of Pelagia panopyra, which is from the Indian Ocean. So that was quite interesting. I was really excited to see that. Other trends, though, were quite similar to what other studies have published. So, for example, specimens from the Bay of Bengal, Indian Ocean, was placing mostly basal to all the other taxa, and also formed a quite distinct cluster. You can see that at the bottom of the phylogenetic tree over there. And a lot of the Mediterranean and North Atlantic species obviously tended to cluster together as well. We know this from the work by Stopa and Ale from the past as well. Costa Rican samples also formed a distinct cluster. You can see that over there. Again, this is not really new. But what was really interesting was that there were some distant Mediterranean haplotypes that clustered with some of the Indian / Bay of Bengal sample, so that was quite cool. We need to explore that obviously a little bit further. And even more interestingly was that the North African pelagia that we collected formed a completely distinct cluster, and we still don't really have any idea why. We need to explore this a lot more. Maybe looking at the morphology in a lot more detail and exploring the genetics a lot more as well. But I thought that was very interesting. And this was a cluster of samples from Morocco, Angola, and just outside the Gulf of Guinea. So that was also quite interesting. So it shows that there could be higher levels of structuring that exists along the West African coast, and Pelagia is seen along the West African coast all of the time. And so this is why I've called this hidden in plain sight, because we see them all the time, but we had no idea that we had this level of structuring and diversity existing along the coast. Now I'm going to switch to talking a little bit about Chrysoora, which is the love of my life. So we managed to collect quite a number of Chrysoora from the West African coast as well. Probably one of the more widespread collections that we've managed to make over the last decade or so. And so those of you that are familiar with Chrysoora would know that along the west coast of Africa, we have 4 species that occur there: Chrysoora isosala, Chrysoora africana, Chrysoora fulgida, and Chrysoora agalensis, which is a recently erected species. Interestingly, I did not recover any Chrysoora isosala in any of the samples that we collected. This blew my mind because traditionally this has been thought to be the dominant species along the west coast of Africa. I saw no Chrysoora isosala. I did, however, recover 2 morphological specimens that was phenotypically quite similar to isosala, but when we looked at the genetics, we only saw something in the region of about 2.4 to sometimes more than 3% variation, but that was sort of where it stopped. So the variability was quite low. So just diving into this a little bit deeper, just a simple phylogenetic tree again. We obviously didn't recover a whole lot of Africana, but the Africana that we did recover also showed some very interesting trends. Again, North African samples were basal to all the other samples, particularly the Western African samples where Chrysoora africana is typically collected and recovered from. So that to me was also very interesting. There were also some differences in phenotype. And one thing I found really interesting was the further north we collected Africana, the more and more it started to resemble Chrysoora pseudosolata. So that was quite interesting, and I'd like to unpack that a little bit more. And we also saw high levels of genetic variation of up to 4% between the North African samples and the Southwest African samples, which was also quite interesting to me because it shows that there's something going on in the north of Africa that we need to explore a little bit more. Another underexplored, very underexplored region. Now, the Chrysoora fulgida gave me a headache. Every single gene I analyzed produced almost a complete star tree, and I freaked out because I thought I was doing something wrong. What I've presented here is the best resolved tree that I could recover. No matter how many runs I did, this was the best tree that I could recover, and you can see all the support values are incredibly low. But I'll come back to why I think we were seeing this in just a second. But the one thing I do want to point out is when I dove into the genetics a little bit deeper, I found that across the entire Benguela region, spreading up to Angola, every single sample that we collected, when we compared them, showed less than 1% pairwise sequence divergence, which I didn't really expect from the region. I thought there would be a lot more variability, but it was coupled with extremely high levels of structuring. So we had FST values that were incredibly high across the region, but coupled with extremely low diversity, and that was quite interesting for me to see as well. I'm going to come back to these 2 C. isosala specimens if I have some time at the end. But just to say that we did also see a lot of morphological variability in the specimens. The specimen on the far right is a juvenile specimen, so obviously we know that they look quite different. But I just put a couple of images up there so you could see some of the variability. Not too variable, but there was some variability that we could detect. So here's where things get even more interesting. Chrysoora agalensis is a recently described species, and we presumed a recently diverged species. But when we started looking at more morphological specimens, more taxonomic specimens, more genetic specimens, we actually started to see kind of high levels of structuring that we didn't expect in something that was supposedly a newly diverged species. So that was very interesting for me to see. And again, large variation in phenotype. These are all coming from the exact same bay. And you can see all the variations that we can see in terms of the phenotype. There's lots of variation, and I have absolutely no idea why this is happening. But the more we sample, the more phenotypes we're starting to see, and we see lots of structuring even within the same bay, in the genetics, but also in the morphology. So that has also blown my mind, and I'd like to really unpack that quite a lot more. I won't have a lot of time to talk to some of my next results, but what I thought I would highlight here, although I'm not presenting it in the slides, is that I did also obviously draw a bigger phylogeny that incorporated all known Chrysoura. And what was very interesting was that this species was basal to Chrysoura fulgida in every single phylogeny, which might suggest that actually this is the ancestral species or the source population. And then all the sort of star topologies that we're seeing in the Chrysoura fulgida might make sense, because it might be indicative of recent population expansion or rapid population expansion. Just a theory, we'll have to unpack that quite a bit more. Now I wanted to talk, oh, my slides went a bit funky. I wanted to talk a little bit about some of the other analyses that we did, but I'm going to put a pin in this in the interest of time, and maybe just highlight that because of the wonderful work that's coming out of the Dawson lab, they've been standardizing a lot of the procedures. And so because of that, I was able to duplicate a lot of Aboud's work across African LMEs, and we actually uncovered quite a large amount of structuring within the African LMEs that I'm not going to talk to right now, but you're welcome to come and chat to me about it during the lunch break. So I'm going to skip those and maybe just end off by saying ultimately what is really exciting is that we are starting to build a baseline of evidence for species diversity across Africa, which is not really there right now. And though this might not seem like anything pioneering, I think it creates the avenue for better, bigger, bolder questions in the future, and it's helping us uncover the scales at which those questions need to be asked. And I'm hoping that in the future, many more maps look a lot like this and a lot less like the first one. This is a horrible map, I know, but this was horribly distorted when I visualized it. But just to give you an inclination of what I hope to see in the future. So thank you to everybody who participated in all of these efforts. You really made my time in the lab really, really wonderful. Thank you.